So we'll get started.
Yeah, I think people will start coming in like later on if they have extra time, but.
Okay, thanks for Justin's invitation. My name is Paul Lu. I work at University of California, San Diego. That's where Justin's hometown. Yeah, Justin did some like summer intern in my lab, and he did very well. Now he's in your university, and I'm just going to share because you are in. I know your background is bioengineer and a graduate student. Just share one of a small story in my previous work. It's involved some bioengineer concept. So the topic is bridge the injury spinal cord with neural stem cell. So yeah, spinal cord injury, it's one of the nerve system injury. It's a central nervous system. It's damaged the local spinal neuron, but that's acted, it's not have severe consequence. The severe consequence, it's also damaged the white matter axon. It's active, it's a nerve fiber travel in the spinal cord. The spinal cord meaning it's bundles of nerve fiber travel. And then when that fiber was damaged, was cut, and this communication between brain and the rest of the body get disrupted. And that's where it causes the paralysis. Actually, you will know my story pretty soon. I'm also a spinal cord injury. That's how I was involved in spinal cord research. Yeah, the big issue is that there's no spontaneous regeneration. That's the character of central nervous system. Peripheral nerve regenerate, and most tissue on our surface, especially the skin, regenerate very well. So we get damage, it's get repaired automatic. But nerve, it's not. We don't know why. There's some hypothesis. It's because the environment is not good, extrinsic factors, but people think also it's their like molecular regulation. It's once the neuron is mature, get connected, their regrowing program, it's stopped. So the big challenge is that it's a big physical injury cavity formation. So after injury, this like this one, it's a T10 crush. You can see after crush, this is maybe 1 month later, you can see there almost most tissue is gone, very little left. And it's especially the neuronal tissue, it's gone. And so there's no way for regeneration because if there's an empty cavity, and there's another story is that in the nerve regeneration story, if you cut the nerve, like I don't know if you can see my arrow. If you cut the nerve,
you can, yeah.
And it's not simple. You just rejoin them together. It's like wire. No, actually, the distal part, it's degenerate. Let's go back. This is called vulnerable degeneration in biology. So once the nerve fiber is cut, you need something reconnect them. It's not a simple connection, replace the degenerate part. So for example, here, this figure shows a tract of nerve fiber. This is the injury. It just stopped here. And anything below this injury, it's gone. It's completely gone. So how to repair this? The best way act, it's the naive neural stem cell, because that's where the original building block of the spinal cord. So for example, this one, it's the green cell, it's a neural stem cell transplant, but this is already mature after a month or so. The neural stem cell will become neuron and glial and can replace lost neuron and glial. Most important is that neural stem cell actually also can reconnect damaged fibers. So when we have, this idea exists for a long time. Practically, the big problem is that neural stem cell is very vulnerable in that injury environment. It's not survive well, and people try. The big issue here is that if you have, let's see, this is a complete transection. You can see there's a gap there. When we transplant cell, ordinary cell usually stick together. But when we culture them, or we isolate them from our body, and then we digest, we detach them, so they become a cell suspension. And it's just individual cell like ball, free floating inside this buffer. And just like let's say a rice soup, and there's nothing sticks them together. So the big problem is that when we transplant this cell suspension in an open lesion, it's not stay there, it's flow away. There's a few reasons. The one reason is physically, it's because the bleeding, when you make a lesion, when you open up, you make bleeding. The bleeding flush the cell away. Besides the bleeding, there also have this we call cerebral spinal flow. It's like their central nervous system, its own circulation system. It's also constant flow. It's wash cell away too. And then there's another reason. After a new injury, the blood-brain barrier broken, the vessel broken, the blood cell coming in, a lot of white cell become inflammation. And this inflammation cell also try to clean up any floating cell around. And of course, there, as I said, it's a cavity. There's no, the cell needs something attached, but big cavity. Some cell you can see attached to the edge, but this attachment, it takes several hours overnight to get something stick together. So that's a big challenge. When I was begin to do this transplantation study, people already try. The big issue is that because the cell not survive in the lesion center, and people grab cell above and below the lesion, but the injury part is still empty. So it's not too much useful. So I'm just thinking, we have to solve this cell not survive, cell not stick together issue. Is there anything that can hold the cell together? And then I search the literature, but actually in the blood system, there is such protein, it's called fibrin, can hold the cell together. And so the basic story is that when you get wounded, the vessel broken, it's called clotting factor. Actually, it's called thrombin. The blood contains a protein called fibrinogen. And fibrinogen, it's like a monomer. It's soluble. But when the thrombin come in, this fibrinogen become polymer, and then that become a gel. And then it's become insoluble. That's how our bleeding get clot, and then the bleeding stop. And so you can see this above figure, blood cell surrounded by fibrin. The fibrin is a net that holds the cell together. When I see this picture, I said, nature have such a wonderful thing. This is what exactly we need. We need this fibrin to hold our cell so they can stay there, they can survive. So actually, when I look at the more literature, and people already use this fibrin scaffold as a glue. When like a medical surgeon did some surgery, so they use suture to suture the let's say the cutting together, but it's not completely stick together. So they put this glue. It's two component, as I said, fibrinogen and thrombin in the different tube. But when you mix them together, within a few minutes, 1 to 2 minutes, it becomes gel. And then it's sealed. It's as a glue for surgical. I said, that's perfect. So what I did is that I get these two component fibrinogen. I put a neural stem cell. Not only neural stem cell, I think neural stem cell survive also need growth factor. I put a growth factor with fibrinogen. Then I inject this fibrinogen component into this injury site. And then immediately I inject this component, then I inject the thrombin with neural stem cell and growth factor. And within a few minutes, they form gel. And they hold the cell together. The cell will stay here. So we have so this is a great successful act. We have a small video I want to share with you. You can see.
In just a few years, custom-made replacement body parts built from a patient's own stem cells will be a reality. But these two men want to push stem cell technology even further.
If they succeed, that would be a profound achievement. One that would mean a great deal to me personally. Can stem cells cure paralysis? Our bodies rebuild themselves every day. We create millions of new skin cells. We regenerate our muscle fibers. Slowly, we are beginning to understand these natural repair mechanisms and to manipulate them. But some parts of the body don't seem to have any ability to repair. The nerves in my spine have been slowly degrading since I was in my 20s. No one has yet found a way to regenerate them.
But Paul Liu and Mark Tuzinski believe stem cells will help them succeed where all others have failed.
Mark, I find one.
Well, let's see. Okay. All right, let's give it a go.
Okay, let's go. 16 years ago, I had a terrible car accident. It broke my spine, and I was desperately looking for medical research can cure the spinal cord injury. And that's how I find Dr. Mark Tuzinski. I write him a letter to request if I can work in his lab.
So we met, and I was really struck by his dignity, his intelligence, his potential. And so Paul joined the team.
Reconnecting a severed spinal cord is like rebuilding the electrical system of a wrecked car. Only 1,000,000 times more complex.
So this is our cut spinal cord. You can see we have about 30 cut wires here, but in reality, the spinal cord has about 1 million. have to connect each one of those from the right spot where we've done the cut to the right target a long distance away.
Fixing a car's electrical harness is straightforward. Solder the cut wires back together, and the electricity will move along them again. But in a severed spinal cord, every nerve below the cut has to be regrown from scratch.
In the real spinal cord, you have to do this 1,000,000 times, from one right one going to the other right one, but all these wires go away. You have to put in cells here that will grow new wires and link them up to the right targets. This is an enormously challenging task.
Paul thought injecting stem cells into the injury site could automate this intricate rewiring process. But Mark was skeptical.
And I said, you know, Paul, people have been working on that for 100 years and, you know, it just hasn't gone very far. And so Paul basically went off and did some experiments and brought back some results, and they were absolutely astonishing. These cells that Paul had implanted, few survived, but the few that did sent their wires, their axons, for remarkable distances through the spinal cord. And this was, in a sense, the holy grail of spinal cord injury research, to be able to grow axons for long distances.
But both Mark and Paul knew that getting stem cells to change into nerve cells and then grow long axons was only half the battle.
For a spinal cord, especially for severe spinal cord, it's a big lesion cavity.
A key step then at that point was to fill the injury site, not have a few cells survive at the edges of the injury, but to fill the lesion site so that more cells survive and can send out more axons.
Paul and Mark decided to use a protein called fibrin, which forms a mesh over the injured area. They hoped it would create a foothold for the stem cells to latch onto.
Then this amazing phenomenon happened. Almost all our graft stem cells survive.
I took a look into the microscope. I backed away my chair. I turned to him and I said, "Congratulations, I've never seen anything like this." The injury site was full. It was glowing green with surviving cells that completely filled the injury. And yet more astonishing, there were now tens of thousands of axons streaming out of the injury site for very long distances. And this in the most severe type of animal model of spinal cord injury.
This is the proof that Paul and Mark's work is actually healing spinal cord injuries. This rat was once paralyzed in its front right leg. Now, it can pick up food with it. This rat was paralyzed in one of its hind legs. Now it can walk across the obstacle course of this cage.
It's not complete recovery, but that is a huge amount of recovery after an injury as severe as that.
This is just a beginning. It shows a potential. We still face a lot of challenges. Like, can this wire connect to the right target? We have good hope.
Mark and Paul have shown that the biological fortress of the spine can be conquered, that stem cells can grow any tissue, anywhere.
In just a few...
Okay, so that's a video with Stephen Hawking. You may know him.
That's quite amazing, I have to say.
Yeah, he has this ALS. It's like motor neuron paralyzed. But anyway, let me show you a few slides about the result of this fibrin. So this is a contusion injury at T10, at thoracic 10 level. And this is a graph. We saw a lot of fiber growth is that we use green rat as a transplant. So we can see what's happened to the transplant cell. The transplant cell is green, so we can monitor it. And So this red, it's neuron. It's a neuronal marker. You can see the graph become many, many neurons and reconnect this part. And the blue, it's a glial, it's a supporting cell for the spinal cord and brain. It's also reconnect. So our graph completely bridge the injury site from this big cavity to complete full. And amazing story is that once this neural stem cell survive, fill the lesion cavity, as in the video show, we have this massive, aggressive nerve fiber grow out from the graph. And this is automatic. We didn't do anything. As long as they survive, they have the full capacity. They follow, because this is a developed, the early stage neuron from the neural stem cell. They have the full capacity to grow in the adult injury environment. And this is, you can see how much new fiber to grow in the white matter, but also in the green matter, make a connection. Another amazing thing we didn't expect is that this graph neural stem cell also attract host axon regeneration. And this red, it's called corticospinal. It's our brain from our cortex in the brain have a bundle of axon travel to spinal cord, control our volunteer movement. And they're robustly regenerate into the graph. And this is actually, it's a large cervical bilateral contusion with a large lesion, and the graph survived perfectly. And you can see the regeneration of this corticospinal tract axon. So basically, our whole story is that once the graph can bridge the injury, and the graph derived neural stem cell neuron function as a relay. It's called neuronal relay. It's meaning host axon, that's it, from the brain, this red, regenerate into the graph, make connection with graph. And graph neuron automatically extend their axon into the host and make a connection to the neuron below the lesion. So graph the neuron function as a relay, reconnect the spinal cord. That's so far, it's, I mean, we have some evidence. That's our basic hypothesis, and we try toward this direction. And we have evidence to show there are some certain functional recovery, certain physiological, we can do some electrophysiology to test this hypothesis. So yeah, just a short summary. And so spinal cord, this is neural stem cell can bridge the injured spinal cord. I write it wrong. It's basically, it's a neuronal relay formation. And as in my video, I said, the big issue is now we have the reconnection. The only issue is that this reconnection might be random. It's not, because in the development, in the early stage of our life, this nerve fiber grow, we have this guiding molecule guiding them to certain direction, to certain location. But in the adult environment, this guidance molecule, mostly it's gone. And in the development, and they produce different kinds of neuron, they will coordinate each other and make the right circuit. But when we graph this neuron, it's more random. And we're thinking, use bioengineer, use scaffold, and try to also give them a spatial arrangement in the right order. So we are currently doing this kind of work. And also the graph axon, graft-derived, this nerve fiber have the full capacity to grow. But host axon, although I show cortical spinal tract regeneration, it's robust regeneration, but other system we tested, it's not that robust. The host axon still not regeneration very well because their intrinsic character, I mean, they're already shut down their regrowing program, and we need use some molecule technique to reboost their regeneration, regrowing program. And another important part is that even you have all this new wire, even the wire is connected right, you need to train this wire to be functional. Just like our baby, it takes a year to learn how to walk, to stand up, to walk. You have the wire, but you need practice, you need work. And usually in our research, we ignore this part. And but also because we use mice, rat, the rehab training, it's not easy, not as easy as human being. But anyway, so the basic story is that we use neural stem cell with this fibrin, get successful, survive, and axonal extension, reconnection. This new circuit, it's there, and we need just optimize their connection. We need to training them. So it's just a short story, and in the video acted, it's my mentor, Dr. Mark Tusinski. This is him, and he gave me the opportunity. As I said in the video, I was spinal cord injury and desperate, looking for someone to do the research I can contribute by myself. So I worked in his lab for more than 20 years until someday I'm retired. But just hope this research will be continued by next generation like you guys, and have new idea, new technology, and continue this research. So, I mean, this spinal cord injury is one of the neurological disorder, but there are many other disorder, many other neurological disease, and of course, other disease too. Hope, I mean, you guys learn something and then have a way to face the challenge for other medical problems.
Thank you.
Okay.
I assume if there are any questions, go ahead. Maybe there's just a couple minutes and take a look at some of the, just ask some questions about like any presentations if you guys have any. I have some of my own, but I'll let you guys go first.
Yeah, I certainly have questions. Thanks again for joining us, Paul. And that was an amazing presentation. I truthfully didn't realize that that stem cell research had reached this point. I thought that axon regeneration was still effectively impossible, but that was incredible to see. I was wondering, in the animal models, have you had any problems with immune response?
You mean immune response?
Yeah.
Yeah. So that's another story. Basically, in the animal model, we use an ideal model. In the rat, in the mice, we have this strength called inbred, meaning you graft cell from the individual animal to another individual. There's no immunorejection. Actually, the real stem cell research or clinic usage, we face this problem. If you graft a neural stem cell from one donor to another patient, you need immunosuppression. It's just like an organ transplant. You have to give the immunosuppression. That's a big challenge. But in the stem cell field, like you guys heard this induced pluripotent stem cell, iPSC. I mean, in theory, you can transplant patient's own cell to themselves. And in that way, there's no rejection, no immune reaction. The cell should survive well. So that's research. It's very active going. I mean, this induced pluripotent stem cell, it's a revolution for the regeneration, medical regeneration. It's Yamanaka, Japan in 2007, 2008, published papers, and he got Nobel Prize 2011, 2012. So yes, so we work on this.
That all makes sense. Thank you.
I was curious about the Axon Direction thing, because I mean, you mentioned in presentation that again, you need to make sure that you have specific direction in terms of where the connections are being made after you implant the graft. Have there been any developments in that thus far, since it's been a bit?
Yes, so we're learning from the development. It's called guidance molecule, guidance axon to the right target. So actually in the nerve system, it's a very interesting phenomenon. It's a target neuron, a target, a muscle target. There's secrete molecule, it's called neurotrophin. And this neurotrophin attract axon to grow in that region, make a connection. Once they make a connection, this neurotrophin can retrograde transport to the cell body, support their survival. But in the development, in the beginning, there's a competition. We produce more neurons than we need. They compete for that target. The one with the real connection, they survive because it's neurotrophin, this guiding molecule, neurotrophin, transport to their cell body, said, you are okay, you can survive. But the next neuron without connection, it just die. So actually in the development, we have like at least 50% neuron die because of competition. So we know these guiding molecules, we can apply, we can like use virus to transduce this gene into the target, like motor neuron, and then to guiding this axon to this region. Like for example, our hand control, it's by cervical 7, cervical 8 spinal cord, and we can specific target that region with this neurotrophin, and to see if they can attract, guiding this molecule, the fiber to make the right connection. Yep.
I was also curious, do you think there's a limit to, for example, the extent of time post-injury that regeneration would still be achievable, and especially connection to the host neurons?
Wow, that's a great question. In that case, it's called chronic spinal cord injury. So chronic, there are more issues, and we work on this project too. The big issue is that in the spinal cord, it's similar to other injury, and there will form a scar. It's called glial scar. And glial scar, a lot of people think it's a physical barrier, and we need to overcome this physical barrier. There's some molecule can dissolve this scar, and but in our study, actually, the neural stem cell also halfly digests that scar, so makes a good connection. But there's still some barrier between graft and host still. So that's some challenge. But other challenge like the chronic spinal cord injury, it's usually also, the regeneration program was shut down more. So you need to give additional factor to stimulate them to regenerate. But anyway, most people focus on the acute, immediate injury. We'll do this first, it works well in the early stage injury, then we'll transfer this at new way for the chronic injury. But
Interesting.
yeah.
Yeah, thank you.
Any other questions? Okay. If not, then I assume that this is being recorded, I think. Well,
Yep.
yeah. So we can probably post that like on our end, and then maybe if you have any questions, you can either forward them to me, or I think I can forward them to Paul, but I think that on the UCSD website that I also sent out, his contact information is there. So I don't know if that would be okay with you, but if anyone
It's
else has
okay.
any extra questions, then yeah, that is also an avenue.
Yeah, and
No
I
problem.
also know that there were some people who wanted to attend but couldn't make it at this time, so they'll get to see the recording, and then I hope it's all right if they are also able to contact you with questions they might have.
Okay, that's great. No problem. I mean, it's a good opportunity for me too. I mean, to pass knowledge to the young generation. I mean, the most important is inspire you to do biomedical research to solve this medical issue challenge, and that's the whole basic concept of the higher education research. And yeah.
This was really inspiring, so thank you again.
Okay, you are welcome.
I would say on a personal note, I have not seen that video before, and that was a really amazing video. I like that video a lot.
Yeah, that was incredible.
Okay, that's great. That's great. Actually, I mean, it's just to know, because that story, it's a small technique improvement, but the result is astonishing. We published a paper in the Cell in 2012. Have a great impact. I mean, I don't know if you guys know it or not, like in biomedical research, I mean, general research, biological research, the result will be published in a journal. There are many journals, and some journals have very high impact, meaning it's a lot of people read it, and a lot of people cite that paper to write another paper. They use that technology. Also check. So the journal have rank and very high journal, high impact journal, lower or general. Cell, it's one of the very high impact journal to publish. I mean, I'm honored to publish paper in the Cell. It's achievement. I mean, if you do well, and people will recognize you and give you like honor. That's how, it goes. But it's also in the biomedical research. You have a good idea, you make achievement, and the government agency, the private foundation will give you more money to encourage you to do that research. So
which always helps the research itself too.
Yeah. So it's a good feedback. So we did well, and it's opened a new door. I mean, like before, people just didn't successful very well because it's persevered. We don't see the potential. But my research is to see that neural stem cell almost in the full potential. And we didn't realize before, but with this idea, and also this green, I mean, before in like early 2000, there's almost no green fluorescence protein marker. So, but we use the green fluorescence protein technology. So we can monitor each cell graph, what's going on. So it's new technology. It's just in the biomedical research, it's changed. It's similar to like computer science.
Yeah.
I mean, you have update, you have this program speed go up. Similar biomedical, it's slower, but but similar. New technology involved every several years. There's a new way. There's a new tool to do research. So it's just getting better. And our knowledge expanding, we know much more. And we have the new tool.
Yep.
That's how you guys study in the university to get this knowledge and to learn this new tool for the future.
Yep.
Okay.
All right.
If
Well,
there are no other questions, yeah, then I think we will let everyone go. Thank you so much for doing this. This is a very fun presentation. I mean, again, I've seen it, like the general idea, but it's still really awesome every time I see it. So, and I hope that that's similar. Yeah. And I hope that that similar sentiment was carried by everyone else. Here.
Okay.
yeah, absolutely.
Okay. Great. Okay.
Yeah.
Thanks, you guys too.
Yeah, thank you
Okay,
so
have a
much.
great rest of the day, everyone.
Yeah,
Bye.
I might see you, Paul, in a couple of days. So
Okay.
you might drop down and say hi. Thanks so much.
Bye.
Bye.